Kraw Lecture. My name is Alex Wolf, and I'm the Dean of the Baskin School of Engineering. Unfortunately, George and Rafe Kraw are not able to be with us tonight, so I'll be George. I'll try to be George. George and Rafe created this series, lecture series, to showcase the highly regarded and impactful research conducted by UC Santa Cruz faculty and students. As some of you know, we're celebrating the 25th anniversary of the Baskin School of Engineering at UC Santa Cruz. Anniversaries like this afford us a moment to reflect on our accomplishments, and we have very many of them, like our pioneering work in genomics, AI and machine learning, computing, human-computer interaction. The list goes on and on and on. But what excites me the most is the promise of the next 25 years at Baskin Engineering. Innovation is accelerating. We are, in our lifetimes, will be the beneficiaries of many important new technologies. Things are changing, frankly, under our feet. Unless we stay in touch with those, unless we stay ahead of those technologies and technology changes, as a society, we'll be in a very difficult place. So I'm very proud of the work that our Baskin Engineering faculty and students are doing to help society meet the challenges of the day. And I can't think of a better representative of that than our speaker tonight. So Daniel Kim is tonight's speaker. This is actually his second Kraw Lecture, and he really embodies the promise of that next 25 years of future. It's very likely that everyone in this room and listening online has been touched by cancer in some way. Now imagine a simple diagnostic tool that can detect cancer early in the most preventable stages. Daniel Kim, an assistant professor of biomolecular engineering, and his team are investigating how the dark matter of our RNA may unlock precision health for us all. His research has been featured in the media and has been supported by awards from the American Cancer Society, Damon Runyon Cancer Research Foundation, Cell Press, and the National Academy of Sciences. So I don't want to take any more time away from Daniel, so I'll ask Daniel, please welcome and let us enjoy the fruits of your research. Daniel.
Great. Thank you so much to Dean Wolf. It's really a pleasure to be here with all of you. Thank you for making time out of your busy schedules to come and hear about some of the work that we've been doing in the Baskin School of Engineering in our lab to really enable precision health for all through the power of RNA. And I don't have to really tell you about the power of RNA, since we've probably all heard about the COVID vaccine, the mRNA vaccine, and how powerful that's been in terms of being able to help people prevent disease. And so really today I'll be telling you about not using RNA so much as a therapeutic, but really how can we leverage the power of RNA to interrogate the health of every single organ and tissue in our bodies. And so if you'll indulge me a bit, imagine if you were to go into the future a few years. So for example, if you had like a DeLorean, for those of you who are too young in the audience, a DeLorean is a time machine. It's like a really fast car that if you hit 88 miles per hour, you can go to the future. But imagine if you were to go to, for example, your primary care physician for your annual checkup. And typically when we go, we draw some blood, right? And we get our cholesterol levels. Maybe we should cut back on some of the in and out or things like that. But really, what if in the future there was a way that instead of just getting a few different measurements, for example, like your LDL or HDL, what if there was a way that you could really get precise molecular level information at a very comprehensive level for all of the tissues and organs in your body? And so here, if you look at the top right, there's a little diagram of DNA, and really this is enabled. What we're doing right now in the lab at the Baskin School of Engineering is really enabled by work that has been done by my colleagues over the past few decades, particularly David Hausler, who has really been instrumental in sequencing and analyzing the human genome, and also some of my other colleagues, such as Benedict Payton and Karen Miga, who have really enabled us to have a much more comprehensive view of our genomes. Today what I'll be telling you about is not the DNA, but really what is the output of the DNA, and why is that important for understanding human health? And so, in the future, let's envision a world where, again, you go into the doctor's office, you draw a tube of blood, and you can really sequence every single RNA molecule that's circulating in your blood. And so if we were to just take a tube of blood from any person in this room, even today, we have the capability, we have the technology to generate libraries, is what we call them, and we could sequence them and understand what are these RNAs that are coming from the different parts of your body, and what does that tell us about the health of your organs? What does it tell you about whether you're at risk for cancer, for example? And so, this is, even though it looks very futuristic and very, sci-fi in a way, really we have this capability today. And so, just again to remind you, the human genome, of course, has 3 billion plus base pairs. And the human genome, of course, is really interesting in the sense where it encodes all of the genes that make our cells and that make the body. But really, when you look at the output of the genome, about 75% of the human genome generates RNA. And so here, if you look at this diagram, the DNA is indicated here, the double-stranded in the gold, and then that's transcribed into RNA, which is, of course, made into proteins. And so if we think about RNA, we really want to understand how does this kind of intermediary molecule really inform just the health of our bodies. And so, again, this is kind of a question that our lab has been trying to address. How will this discovery into our transcriptomes, the complement of all the RNAs in the human body, how will that really enable us to live healthier lives, really to be able to diagnose disease before it is too late, before it's, for example, spread throughout the body. And so we really focus initially on cancer because we know that cancer early detection has been shown to save lives. And in order to achieve precision health, we really want to be able to diagnose disease very early before, again, it's spread to other parts of the body. And we know that cancer is a huge and challenging problem, as we all know. I'm sure everyone in this room has been touched by cancer in some way. And we know that, for example, if we look at the number of new cancer cases worldwide, there are over 18 million cancer cases currently diagnosed, and there are almost 10 million cancer deaths worldwide, and this is on an annual basis. And for example, approximately 40% of men and women will be diagnosed with cancer at some point during their lifetimes. And if we kind of look at the global statistics again, here you could see the estimate in 2020 was that there were about 18 million cancer cases, and this number will only increase in the coming years. And so by 2040, it's estimated that there will be about 27.5 million cancer cases. And unfortunately, this leads to more increase in cancer deaths. For example, in 2020, it was estimated that about 10 million people died from cancer, but this number will continue to go up. And the reason why people die from cancer is typically because these patients detect this disease very late in the process. And so about 90% of patients die from metastasis when the cancer has spread from the primary site of origin. And typically this is due to late diagnosis because only during these later stages do people start to get symptoms. They go to get checked out and they realize that it might be spreading to other parts of the body. And there's another pressing issue, and that's the fact that we only have 4 recommended population-level cancer screening methods currently in the United States. And so for the vast majority of cancers, of which there are upwards of 50 different cancers, there is no good screening modality. And even if you think about the existing screening modalities, for example, for breast cancer, there are mammograms. For colorectal, there's colonoscopies. For cervical cancer, pap smears. For lung, there's low-dose CT scans. But if you think about it, if we really want to make an impact in terms of being able to diagnose cancer at the earliest stages, there's really no way to know which of these screening tests will be effective for any individual. And so if someone really wanted to prevent any kind of cancer, they would first of all have to get all of these screenings, except for, of course, for these 2 or more female specific. But even if you were to do these screening modalities that are currently recommended, you would again miss the vast majority of cancers. And so there is this really pressing need to do this kind of multi-cancer diagnosis. And so there are a lot of people in the Silicon Valley, as well as throughout the world, who are really trying to develop these non-invasive approaches called liquid biopsies for cancer early detection and for multi-cancer early detection in particular. And so essentially what a liquid biopsy is again, if you were to draw a tube of blood, you could sequence all of the information, for example, all of the DNA that is being shed from these cancer cells into your blood, and to really try to detect cancer at the earliest stages. And so typically what people do is, again, they draw some blood, and then they look for this signal, this circulating tumor DNA signal. The problem with that is, even though there have been a lot of efforts, a lot of very well-funded companies, for example, have been pursuing this, what they have shown based on these very large-scale studies where they've looked at thousands of different patients over time is that these DNA-based tests, these DNA liquid biopsies, actually miss the vast majority of early-stage cancers. And so there's one particular company, which I won't name, where they have done this DNA-based test, and they've shown that, for example, for stage 1 cancers, they miss these stage 1 cancers about 80% or more of the time. Sometimes it's about 90%. And so really, to be able to enable early detection, we need to do better than missing, for example, 80% of the early stage cancers. And the reason why the sensitivity is so low for DNA is really biological. If you think about a cell, for example, let's say this is a kind of cartoon version of a cancer cell, the DNA is packaged in the nucleus, right? And in order for that DNA to get into the blood so that you can actually detect it using a liquid biopsy, the cancer cell actually has to die. So it has to die, rupture, and release its DNA contents into the circulation. The other issue, though, is that once the DNA is in the circulation, it degrades very rapidly. So the only pieces of DNA that actually survive in the blood are the pieces of DNA that are wrapped around what are called nucleosomes. And so these are protein complexes that about 150 base pairs of DNA can wrap around. And so if the DNA is not wrapped around this nucleosome, it'll actually be degraded, and you'll never see that signal. And so not only are you fundamentally limited by the fact that the cancer cells have to die to release that DNA, you're also hoping that whatever DNA does make it out of the cell is actually going to survive the hostile conditions in the blood. And so that's very problematic for early detection. And even with the latest sequencing advances and technologies, and this is the current state of the art, we're again missing over 80% of these stage 1 cancers because there's just not enough information in that tube of blood. However, RNA is very interesting because if you think about RNA, again, I showed that earlier diagram where the DNA is transcribed into RNA. If you have, let's say, 2 copies of any gene, that's going to be transcribed into many more copies of RNA. But the other really cool thing about RNA is that the cell doesn't have to die. It doesn't have to rupture and release the RNA. The cells actually actively secrete RNA in what are called extracellular vesicles. So these nice little balls here, these are vesicles, such as exosomes, that are released from all cells in the body, but particularly they're more preferentially released from cancer cells. And so what we've discovered in the lab is that, for example, if you take a cancer cell and you compare it to a healthy cell, there will be not only more of these vesicles that are being secreted actively from these cancer cells, and they don't have to die, they just continuously secrete this information, but there's a particular type of RNA that's preferentially secreted in these extracellular vesicles called non-coding RNA. These are RNAs that don't make proteins. And so based on some very fundamental basic science that we were doing in the lab, we were able to identify specific types of these non-coding RNAs that were being preferentially secreted by these cancer cells. And so here, now you're not limited by the fact that the cancer cell has to die. You're also not limited to the fact that there's only 2 copies of any piece of DNA, and you're also not limited by the fact that the RNA is going to be degraded, or sorry, the DNA is going to be degraded in the blood, because when these RNAs are secreted in these vesicles, the nice thing about the vesicle is that it actually protects the RNA from degradation. And so we've actually used some technology that was developed by our very own Dave Deemer, my colleague in the BME department, where you can actually sequence individual molecules of these RNAs, and you could see what is the actual true length of the RNA. And what we were able to see is that when you take these vesicles from these cancer cells, for example, and you look at the RNA, it's actually intact. It's not degraded. So kind of the misconception in the field is that, oh, all of this RNA is degraded. You're just going to get little bits and pieces of DNA. But when you actually do the experiment and you sequence these RNAs that are being secreted, you see full-length information. And that's only enabled by this particular nanopore-based technology that my colleague Dave invented. And so when we do this, we could see that there is this really powerful signal that's coming from these cancer cells. And the other really amazing thing is when you think about, for example, again, this idea of precision health, you can really think about it in terms of what is the signal that's coming from the entire body in these vesicles. And so here, what I'm showing you is some of the different features of the analytes that are in the blood. And so here you could see, for example, cells that are secreted from these tumors. They break off from the tumors. So these are, for example, circulating tumor cells. There's also bits of single-stranded nucleic acids, double-stranded nucleic acids. And so again, these are rapidly degraded unless they're, again, wrapped around some protein complex. But the really powerful and amazing component that we're looking at is, again, these extracellular vesicles that protect this RNA information. And so the other thing that is really amazing is that, for example, if you think about the human genome, does anyone know how many protein coding genes there are in the human genome? 30,000? Okay, almost. It's in the right order of magnitude. So yeah, it's thought roughly like 20,000 or so, right? But the nice thing is that when you look at the RNA, you're not limited to, for example, looking at protein coding genes. You can actually see that there are many more RNA encoding genes. So for example, if you look at the human genome, it contains over 100,000 of these non-coding RNA genes or loci. And so what you can see is based on, for example, a lot of research over the past few decades, there are many different classes of these non-coding RNAs. And the really quite amazing thing is, again, in the context of cancer and really early stage cancer, these non-coding signals are what are most strongly perturbed. So in a way, you can think of these non-coding RNAs kind of as like a warning signal in terms of what's happening to these cells. And so really we wanted to address this question of, can we leverage this vast non-coding RNA landscape, these over 100,000 different RNAs, there's actually even more, but I won't go into all of those, for this purpose of multi-cancer early detection, to be able to detect any cancer that's in the body using this RNA signal. And so in addition to the non-coding RNAs, when you look at, for example, the RNA, you see that there's also protein coding RNA as well. So you get this information in terms of the non-coding signal that lets you diagnose a disease, but you also get information in terms of what are the different pathways, what are the different druggable gene targets that are also awry in these patients. And so that also enables not just diagnosis, but now what are the right treatments, what are the right drugs, what are the right targets that you can really drug to cure these patients in theory. So when you think about this, you can think about just this non-coding information that's really secreted. I'm only going to show you one piece of data, so if you'll just bear with me. So this is typically what our data look like, and this is what's known as kind of a volcano plot. And so here what you can see is, for example, the amount of change, the fold change. This is in the kind of positive direction, this is in the negative direction. And really this is just a comparison of looking at healthy individuals, looking at the blood of healthy individuals, sequencing all the RNA that's circulating in their blood, and then compared to patients that have cancer. What are the RNAs that are circulating in these cancer patients? And so here in yellow, these are these protein coding messenger RNAs. So you can see that some are enriched in healthy individual blood, and then some are enriched in these cancer patient blood. But what we see is that these non-coding signals, these signals that are not coming from these protein coding genes, are in fact what are most strongly enriched in these cancer patients in their blood. And so we're really excited about this result because now we can look at this information and try to diagnose cancer very early. And so there's a paper that will be coming out very shortly that describes this result. And if anyone's interested, I'm happy to send that paper to you once it comes out. But the other thing that we've been doing with this work, which has really been pioneered by Roman Reggiardo in my lab, he's sitting in the back over there, and he has been really enabling this interrogation of the non-coding transcriptome, looking at these additional features that people just kind of typically throw away when they look at sequencing data. And so building on that, we really wanted to see, can these non-coding signals, these non-coding RNAs, enable not just the diagnosis of cancer, but also provide, for example, positional information. So for example, let's say you see a few of these RNAs in the blood, like you'd also want to know, like, where is this cancer originating from, right? So once you have that information, you could use, standard imaging modalities to see, what is the size of the tumor, can it be resected surgically, can it be treated with radiation, chemo, et cetera. And so to do this, Andy Davidson in the lab, who's also in the audience, he really began to look at all of this existing RNA data that was generated through these large consortia, where what they did was they took all of the known tissues, all the healthy tissues in human individuals, and then they just looked at all the RNA that's in each of these tissues. So what are the RNAs that, for example, comprise the brain or the lung or the liver, et cetera, to identify what is specific in terms of the RNA signal for a given tissue. And so this is based on over 10,000 samples from healthy tissue, but he also looked at, for example, cancerous tissue as well. So this is from the Cancer Genome Atlas, where they looked at over, again, 10,000 different patients, and they looked across 33 different tumor types, and again, sequenced all of the tumors that these patients had to identify what are the RNA signals that are within these tumors. And so what Andy was able to do was he was able to compare, for example, if you looked at a lung cancer sample, like what does that RNA signal look like compared to all the other cancers or all the other healthy tissues in the body. And so he was able to identify these very specific signatures that would really enable this kind of positional identification of where this signal was coming from. And so, with Andy and with Roman and with others in the lab, we're really trying to identify a way to classify cancer versus not cancer using AI or machine learning to enable this multi-cancer early detection. And we're using these non-coding RNAs as features. For example, these cancer-specific non-coding RNAs that are unique to, let's say, lung cancer or pancreatic cancer, to really reduce the feature space to that which is most relevant for not just diagnosing the disease, but also providing that positional information for where in the body this is coming from. And so we're really excited about the potential of this technology because we know that, again, there is an increasing need for early detection of cancer, but we really want to kind of prevent this kind of right side of the graph, where if you can detect cancer early enough, again, at, for example, stage 1, in theory, we would be able to save a lot more lives because we'd be able to detect cancer before it's had a chance to spread, and you could, again, either surgically remove it or treat with specific targeted therapies. And so really the goal is to develop an equitable RNA-based multi-cancer early detection test that can help save millions of lives around the world. And I emphasize this word equitable because currently there is a technology that enables multi-cancer detection, but it's quite expensive. It's about $1,000. And so we really want to develop a test that would be affordable, but also a test that wouldn't require you to have very specific and very expensive equipment or technology. And so there is, again, as I mentioned, Dave Deemer, my colleague's work, where they've developed this sequencing capability that's really handheld. It's like a portable sequencer. I should have brought one in my pocket, but apologies, I didn't bring one. But really, do you have one by any chance? But really, we want to enable this RNA-based multi-cancer detection, early detection test, where you could, again, sequence this literally anywhere in the world. And they've taken this sequencing device even to outer space, I believe. And so this work that we've been doing was recently featured in Newsweek, where the headline was kind of interesting. Holy Grail blood test can diagnose cancer years before symptoms. It's interesting if you actually read the article, they actually say that my student Roman discovered a protein when it was actually RNA, and they actually said that he discovered this gene called KRAS, which is a very famous oncogene. So he didn't discover KRAS. So if you do read it, just kind of keep that in mind. But really, this is the goal, to be able to do this early detection through the blood, non-invasively, and we have the capability to do this right now. And again, as I talked about before, this this is actually just a picture of this nanopore device that was really, again, discovered by or developed and invented by Dave Deemer, where you could now take, for example, any blood sample. You could generate, for example, a library that you could load onto this particular sequencer, and you can generate information, RNA information about all of the different signals that are coming from, for example, cancer cells in the body. And so we've actually started to do this. I think we've done a few experiments now where you can really see the signal very nicely using this handheld device. And the really amazing thing is, like, for example, there's something called a flow cell that you put inside the sequencer, and it's less than $100 for this flow cell. And you can just pop it in, load in your sample, and do the sequencing in real time. And so we're really excited about the potential of this to enable not just early detection, but affordable, more equitable early detection. And again, this is something that could be taken to resource-poor settings where, again, you don't need some academic medical center, you don't need some huge sequencing instrument. You can really do this simply with this handheld sequencer and a laptop. And so with that, I just want to really thank all the people in the lab that really contributed to this work. As I mentioned, Roman Reggiardo, also Sri Lakshmi Vlandi Murali, who's right here in the audience as well. So they really worked together to enable this RNA liquid biopsy technology. And also thanks to all the people in the lab that came as well. And of course, I mentioned Andy's work, looking at all of these different data sets to enable this tissue-specific, cancer-specific signal identification. Also, my collaborators at UCSC, Stanford, and UCSF. Also, we're very grateful for funding from the American Cancer Society, the Department of Defense, NIH, CERM, the TRDRP, Tobacco-Related Disease Research Program. And it's really, all of this work is enabled by just all of the colleagues that we have the privilege of working with in the Baskin School of Engineering and the Department of Biomolecular Engineering. We have also a really great stem cell institute and an RNA center, amazing genomics institute as well. And so really all of this synergy has enabled us to identify these new RNAs that can enable cancer early detection. And this is our lab at Santa Cruz. If anyone wants to come visit and see what we do in the lab, feel free to reach out to me. Happy to show you around. And again, thank you so much for your time, and thank you for just coming out this evening, and I'm happy to take any questions. Thank you.
Thank you. So, thank you, Daniel. Daniel and I are going to just have a little conversation here. If you want to listen in, that's fine. I'm happy to have you listen in. But actually, I would really love to have you ask some questions, and also folks who are watching remotely, we will also be taking questions. Just put them into the chat in the Zoom room, and we'll endeavor to get Daniel to answer the questions. So let me start off by asking you a little bit about, you went quite quickly. There's a lot of information in there, and there's a lot to unpack, and it's really fascinating. So there's a couple of things that I would really like you to explain a little bit more. So you talked about a library, and that library seems to be kind of a key piece of ground information that you need in order to do the diagnosis. Can you talk a little bit more about what a library is and how that's formed, how that's created?
Yeah, definitely. Thank you. So, for example, if you look at RNA, there's a lot of RNA that's floating around, as I mentioned, in the blood. And what we're seeing is that, for example, when you reverse transcribe it and make what's known as cDNA, complementary DNA, you could sequence that on, for example, different sequencing platforms. But in addition to kind of sequencing it, the kind of more probably important piece is how you analyze that data. And so when you think about a library, you think about kind of the comprehensive, like, entire collection of nucleic acid information. But oftentimes what people do is when they analyze the data, they kind of throw out a lot of these non-coding regions. So they're only subsampling a portion of the library. They're only, again, looking at these protein coding genes, right? These messenger RNAs that are coming from these protein coding genes. Because for the longest time, the non-coding RNA relevance or importance has not been appreciated in terms of cancer diagnosis or just even understanding the biology of the disease. So when we make these libraries, like we have all this data that's generated, right? Like you get all this information, but when you again throw out all of these reads or these sequences that don't align or map to your complement of protein coding genes, you're missing a lot of information. You're throwing out a lot of signal. And so that's where looking at the non-coding parts of the genome is really critical. And so as I mentioned, Roman has developed this way to look at a lot of different non-coding features in the genome, these transcriptomic RNA features. And so that's what really enables us to see really good performance in terms of sensitivity and specificity of these diagnostic approaches. And so I think that this library aspect is critical because we want to comprehensively interrogate everything, right? And we don't want to leave anything on the table in terms of like what we're analyzing. And so there's even more information that we're currently not looking at because there's just a lot of information. But the reason why, again, that early detection doesn't work well for DNA, for example, is that you're not getting this full for example, 3 billion base pair genome, you're only getting what's, again, surviving. Whereas for RNA, like we don't have that issue. We have like over 2.2 billion base pairs that are made into RNA, and all of that will survive if it gets secreted from the cancer cell. So we have all this information that we can leverage. When we make the library, we could sequence everything and analyze everything. And so that's really the innovation, is looking at these non-coding parts of the genome.
So it sounds like the library is comprehensive.
Yeah.
How representative is it of any particular individual?
Yeah, that's a great question. So what we have discovered is that when you look at, for example, the cancer cell and all the RNA that's made in the cancer cell, and then all of the RNA that actually gets exported or secreted from the cancer cell, it's not a complete representation of everything that's in the cell, but there is this bias for these non-coding signals, which is, again, why we're focusing on the non-coding RNAs, because they're preferentially being secreted. So it actually gives us a really strong signal in terms of what's cancer specific. And so even if we don't get the full transcriptome, the complement of all the RNAs within the cell, the signal that we do get is very informative for diagnosis.
But from individual to individual, the library is representative.
Yeah, so if you look at, for example, and this is work Andy has done and Roman has done as well, where if you were to look at, let's say, 100 lung cancer patients, there is a common signal across all of those lung cancer patients. And when we, for example, look in the blood, we can see, for example, a signature that's representative of those individuals. So the signal, if you have a particular type of cancer or a particular type of mutation, that signal is consistent across individuals. So we're able to kind of use that information to generally diagnose like lung cancer or pancreatic cancer.
Question? Wait, we need to give you a microphone. There we go.
Hello? What was nature's reason for excreting these non-coding RNAs?
That's a great question. I don't know. But there is some evidence out there that these RNAs may be involved in cell-to-cell communication. So, for example, if and this is kind of speculation right now, but for example, if you had, let's say, a cancer cell and it's continuously secreting these RNAs in these vesicles, people have done some experiments where they've shown that these vesicles that contain these cancer-specific RNAs can circulate to other parts of the body, and they can actually reprogram other parts of the body where metastasis can occur. So in a way, they're kind of reprogramming these potentially hostile environments to become more amenable to these cancer cells coming and, in a way, colonizing these metastatic sites, right? And so there is some evidence from, for example, mouse studies that people have done where when you see these vesicles that have DNA, RNA, protein from the cancer cell, that seems to reprogram these distal environments where the cancer cell can then go spread to and form these metastatic sites. In terms of what it's normally supposed to do in a healthy individual, yeah, it's not entirely clear, but the thought is that there is some role in cell-to-cell communication.
Thank you for the talk. There are a couple different features people have been using to detect cancer early. My question to you is, what do you think is the earliest feature? Do you think aberrant non-coding RNA in cancer cells is the earliest feature of cancer, and that's why you're focusing on it? Do you think methylation is coming before that? I'm curious to learn more about what do you think that timeline looks like as cancer forms?
Yeah, that's a great question. So we don't know if methylation or RNA is first, but in a way, like RNA, the way we view it is RNA is in a way kind of telling you the regions that might be methylated versus the regions that might not be methylated. So in a way, when people do these like DNA methylation-based assays, it's kind of a proxy for like what regions are transcribed versus what regions are not, right? So there's even studies that people have done where they've looked at like fragmentation of the DNA that's being released and kind of inferring like what regions might be transcribed or not. But the RNA is kind of that direct readout, right, of what's actually transcribed or what's, open chromatin, for example, right? So it could be that they're both kind of simultaneously early marker, but for DNA, again, like it would have to survive, right? Whereas for RNA, we have that nice protected environment for the RNA. So yeah, definitely. But what we've done is we've done some experiments where we just take, for example, like healthy human lung cells. Well, healthy is relative, but we've just introduced a simple mutation, for example, a KRAS mutation, into these human lung cells. And then we've looked to see like what are the earliest changes. So there are definitely epigenetic changes, but there's also a lot of RNA changes where if you look at the secreted RNAs, there are a lot of non-coding signals that just start to get secreted with this one mutation, this very early driver mutation, right? So I think it's both. There's both epigenetic and transcriptomic changes at the earliest stages. It's just a matter of like what can we detect better at the earliest stages? Yeah.
Yes, question.
Yeah, a question from the chat. Ross asks, if you detect cancer from the non-coding RNAs, how do you determine the positioning in the body or the tissue of origin?
Yeah, that's a great question. Thank you, Ross. So going back to Andy's work, so what Andy did was, again, he took all these different tissues, right? All these healthy tissues, and then all these different cancers. And then he compared, for example, one cancer with all the other cancers and all the other tissues to see what is unique in terms of the RNA signature. I guess I should be looking here. What is unique in terms of the RNA signature for that given cancer? And so that enables us to identify where in the body the signal is coming from, since that RNA signal is very specific to that particular tumor. So for example, let's say you see 10 different RNAs that are only ever expressed in lung cancer tissue, and you see that in the blood, then you can assume that that signal can only come from, for example, the lung. And to confirm that, of course, you can just do standard imaging modalities. But really that comparison of these 20,000 plus transcriptomic samples, that's really enabled us to do this kind of positional identification, kind of like this IP address for where this RNA is coming from.
Daniel, nice talk.
Thank you.
Thank you. I learned from it. I had a curiosity question.
Mm-hmm.
RNA, there's three kinds of main RNA in a living cell. It's ribosomal,
Mm-hmm.
messenger, and tRNA, transfer.
Mm-hmm.
Now we know that these, the non-coding RNA are not from ribosomes because we would recognize the ribosomal sequence.
Mm-hmm.
They're not from tRNA because that's too small.
Mm-hmm.
So they are either from degradation of mRNA or they're being synthesized for a function, as you alluded to, that they're kind of like a hormone. They be secreted and turn on some process
Mm-hmm.
elsewhere in the body. Now, I'm interested in knowing, if you know, whether you can find non-coding RNAs that in fact are pieces of the natural breakdown, degradation of messenger RNA that goes on all the time in the cells. You know, messenger RNA has a lifetime of anywhere from minutes to an hour or so, and then it gets degraded. So there's pieces left over, and those could be coming out of the cell. So do you think we know yet how much of the non-coding RNA is just sort of an accidental product of degradation, or in fact has some functional purpose in the cell that you could recognize?
Yeah, that's a great question. So for the context of cancer, we don't know exactly what the function is for some of these RNAs, but going back to that example of when we just take a healthy human lung cell and just introduce a single mutation in KRAS, this single oncogene, this driver mutation, what we see is that all of these transcription factors that normally silence these non-coding regions all get suppressed. So there's like dozens and dozens of these crab zinc finger genes that all get turned off when this mutation in KRAS occurs, this induction of oncogenic signaling. And so when those epigenetic repressors are downregulated, there's this kind of broad upregulation of these non-coding RNAs. A lot of them come from repetitive elements in the genome, like retrotransposon sequences. And what we've seen is that, for example, these transposable element-derived RNAs seem to induce this interferon-stimulated gene signal in these cancer cells, and that signal is also secreted in these vesicles. And so we don't have definitive data for this, but we can speculate that these, not just these repetitive RNAs, but also these interferon-stimulated gene messenger RNAs could be taken up by cells in the surrounding environment, the niche, for example, and can lead to, for example, inflammation in these surrounding cells. And so in terms of like normal biological function, we're not sure, but in terms of cancer, it seems very clear that this stress that this oncogenic signaling induces in the cell leads to this broad upregulation of these non-coding elements. The other thing that's really interesting is, like, for example, if you take a cancer cell and you treat it with a specific drug, let's say that targets this mutation in KRAS, it also leads to this vast reprogramming of this non-coding landscape. And so what we think is happening is that these cancer cells are trying to survive, and so they're reprogramming their epigenome and their transcriptome. They're turning on all these things to see, can that help them gain resistance or acquire resistance to this particular targeted therapy. And so those are ongoing experiments that we're still doing. But yeah, there definitely seems to be this kind of, this response from the non-coding signal to particular stress, like oncogenic stress. Yeah.
So there are obviously a good number of people here who are quite expert in the room. I'm hearing a lot of Greek-rooted words that I don't understand. So I'll represent the other half of the room and ask a very simple question. One of the things you hear about is the fact that people are producing cancer cells all the time, and that, as our diagnostic techniques increase in their fidelity or precision or sensitivity is probably the best word, that we're detecting, in quotes, cancers that are actually not dangerous in some sense, because we're always producing cancer cells. The body naturally protects us from them. So how are you avoiding that kind of false positive diagnostics in this technique?
Yeah, that's a great question as well. So we're actually leveraging a lot of data that was generated by the Cancer Genome Atlas. And so what they did was they looked to see, for example, for these patients that they collected their tumors and sequenced, like what were the kind of outcomes for these patients, right? So if we see, for example, a particular RNA signature in the blood of let's say lung cancer patients, we can go back to, for example, the Cancer Genome Atlas data, where again, we know for each patient whose tumor was sequenced, we know what RNAs are really high in those tumors, right? And then we also know, for example, like the outcome of those patients as well. So in a way, we can get kind of prognostic information in terms of when we see a particular signature, is this patient more likely to survive or less likely to survive, right? So for example, to your question of like, over diagnosis, or like who should we kind of not treat aggressively versus treat aggressively. Like when you see signals that are predictive of poor survival, like that would indicate that you should probably be more aggressively treated for your cancer, as opposed to if you had a signature that was not prognostic for poor survival. So definitely we're trying to take that information into consideration, where we really want to focus on what are the most dangerous signals in the blood in terms of like poor outcome for the patients.
So the other danger is, I imagine, is that it tends towards the diseases that are more common, and the rare diseases, the rare cancers, we know less about. We know less how their dangers and their outcomes.
Yeah.
I noticed that on the slide there was mention of rare cancers. So how does that figure into the work that you're doing?
Yeah, so for rare cancers, by nature of being rare, we don't have as much information, right? But the fact that we can detect those cancers, right, that's going to be really important, because for a lot of these, for example, companies that are doing these diagnostic approaches, like they want to go after the big cancers, right, like colorectal cancer, lung cancer. But for a lot of these rare cancers, if you're developing a single cancer test, that probably isn't going to translate well to doing rare cancer diagnosis. Whereas for our platform, it's really cancer agnostic, and it's actually disease agnostic, which is why I titled it Precision Health, because as we get more information about different diseases, even like neurodegenerative diseases like Alzheimer's, like when we can figure out like what are the transcriptomic changes that happen in the context of neurodegenerative disease, for example, like we could see those signals in the blood as well, because again, we're not looking for a particular signal. We're just looking at all the RNA that's just circulating in the blood. And so in theory, for rare cancers, for other types of diseases, like we would be able to diagnose what's happening. And so, during this COVID era, we also looked at COVID samples as well. And we see that for patients who have been infected with COVID, they have a very different signature in terms of the non-coding RNAs, and also even the types of signaling pathways that are going awry in these patients, where, for example, now a lot of people might be suffering from long COVID and might not really know like how to be treated, right? If you were to take this technology that we've developed and just sequence the RNA, we could see like what are the particular pathways that are awry in those patients, and potentially pair that with the right targeted therapy to more effectively treat them. So this is really just giving you systemic information where, again, it's not constrained by any disease state. It's really just whatever you know about that disease, you can bring to bear on figuring out how this patient should be treated, or what's the best course of action for them. So we're excited about looking at not just cancer, but also other types of diseases as well, where you could think of new ways to treat and potentially even cure these patients. And then, as we talked earlier, one thing that's really exciting is that, for example, in the context of cancer, people have these like neoantigens, like these unique antigens that are only found, for example, in this individual, where there's some recent studies that leverage the new mRNA technology, the COVID vaccine technology, where you can take these sequences, these RNA sequences that are unique to this cancer patient, you can synthesize messenger RNA for that, and then deliver that back into the patient. And for some of the patients, it could be curative as well, because you're essentially enhancing your immune system's ability to fight your particular cancer, right? So you can make these bespoke RNA-based drugs in a way, based on using this liquid biopsy approach to identify what are the right targets. Then you could generate the messenger RNA vaccine, introduce it back into the patient, and potentially treat them in a very personalized, precise way.
You're clearly somebody who is very committed to bringing this solution, this diagnostic technique and tool, to the world. But you are, let me put it this way, an assistant professor in a laboratory in a university, and it feels like there's a big gap there. So how do you bridge that gap? What do you need in order to bridge that gap? Bring your technology further along to the point where it actually can be used.
Yeah. Thank you for that question. So, I don't know if people know this, but like for for assistant professors in research fields, we spend a lot of time writing. In particular, we spend a lot of time writing grants, asking different funding agencies for support for our research. So a lot of my time and bandwidth actually goes into just writing like proposals, asking people to give us money. And so that really does slow things down because, I'm most excited about doing this discovery research and trying to figure out like how can we help save people's lives with this technology. But when half of the time or more I'm like writing, grants and things like that, it really does slow things down. And so really like resources would be the most beneficial in terms of accelerating the pace of translation for helping patients. And so, yeah, that would probably be the most important thing that would be in terms of like being transformative for getting this to patients more quickly, but also, just having the resources to validate this technology. Like, for example, we don't have a medical school at UC Santa Cruz, but we can purchase blood samples from cancer patients using different companies, and that all costs a lot of money to buy, like hundreds or thousands of samples costs quite a lot of money to do that. And really to show validation, like we would need to do this in a lot of different samples. We would need to do this across many different cancers to show that it really is specific for any given cancer. And so it's really just a matter of like having the bandwidth, but also the resources to do this validation as well.
Is there an opportunity for companies to partner in some ways to bring this technology to the market? Is that another pathway?
Yeah, definitely. So, we're happy to partner with companies who, have the expertise in terms of recruiting patients for clinical trials and things of that nature. So, yeah, we would be happy to form academic industrial partnerships as well.
How do you overcome the subsampling error that could occur if you're, say, a patient who gives a couple milliliters of blood and the flow cell can probably handle like somewhere in the microliters? If I want to make sure that you're not missing my cancer, do I have to pay for more flow cells?
So we've actually not been sequencing super deeply, but we could still see very robust signals. So, we're not doing like nearly what people do for like whole genome sequencing, for example. Like we don't need to get like 30x coverage of the transcriptome. We can do it with relatively shallow sequencing, which is why I'm excited about the potential for like the nanopore technology, because we don't need that many reads, honestly. We probably can get away with a few million reads. So yeah, even if we're just subsampling, we're still getting like a very strong signal in that subsampled population.
Question.
Yeah, another question from the chat. We've talked a lot about the different cancers and which ones we might possibly detect, but why are you focused on blood as the body fluid, and why not other fluids?
Yeah, that's a great question as well. So, yeah, blood is I think most exciting for us because really blood circulates throughout the body. So any signal in the body most likely is going to be secreted into the blood. Whereas, for example, if you were to use like urine, like you'd kind of be limited to more bladder cancer or prostate cancer. If you use saliva, like some oral or throat cancer. So definitely, some biofluids do have their limitations. For example, like if you wanted to do like cerebrospinal fluid, that'd be good for like brain, but obviously you don't want like that invasive spinal puncture, right? So blood is relatively easy to obtain, right? Now, people get it taken all the time at our annual checkups. And again, it really circulates throughout the body. So it's getting information from every organ and tissue in the body. So it really gives you that comprehensive information where other biofluids are limited in that respect.
This is a more open-ended question. Do you think RNA secondary structure can play a role in early cancer detection?
Yeah, that's an interesting question. We haven't really looked at RNA secondary structure. I mean, one thing that we have looked at is, for example, modifications to RNA. So we've looked at, for example, RNA editing, and typically that occurs when there's double-stranded regions, right? So what we have seen is, for example, when we use again our KRAS model of lung cancer, we do see that there are changes in RNA editing to these double-stranded RNAs. That are coming from these, for example, transposable element regions. So, definitely that would only happen on double-stranded RNA. So, so there may be a role for different secondary structure in terms of if, let's say, these repeats are coming from these inverted alu repeats, right? So, yeah, we haven't looked super comprehensively at that, but definitely there's potential there as well.
Does RNA cross the brain barrier, and how do you find brain cancers?
Yeah, so these vesicles are actually quite small, so they're kind of in the nanometer size range. So people have shown that you can get brain cancer vesicles detected in the blood. So they can be anywhere from 30 nanometers in diameter to maybe a few hundred nanometers. But at least for the small ones, people do see signal that's coming from the brain. So for example, a glioblastoma signature, we'd be able to identify that in the blood as well. So definitely there's evidence that you can detect it using this vesicle that's being secreted.
You talked about other diseases besides cancer.
Mm-hmm.
Is there something about, are there particular kinds of other diseases that lend themselves to this technique? Or is, that's the question really.
Yeah. So I think anything
because
that's...
cancer is a particular kind of disease, right?
Yeah.
It has a very particular method of cell destruction and reproduction, but other diseases are not at all like that, right? So
yeah.
What is it that's common?
Yeah, so any disease that has any kind of change in terms of their transcriptome, right? So for example, Alzheimer's disease, there are some studies that people have shown that a lot of non-coding RNAs are aberrantly upregulated in the context of Alzheimer's. So we would be able to see all of that signal. So I would say for many diseases, you are going to see some sort of transcriptomic change in the cells that are in a diseased state, right? Just because RNA is so fundamental to the function of that cell. So most likely, I would say most diseases would be amenable to this. The other part, though, is how much signal would you get? So for example, for cancer, the reason why it works better at later stages is there's just more signal, right? Because the tumor is growing. So depending on, for example, how much signal is being secreted by the affected cells, that could certainly affect the sensitivity of it. So there could be some challenges with different types of diseases, but for cancer, it's pretty straightforward, where you're going to see a ton of signal at late stages, but we also see a strong signal at stage 1 as well.
Okay. Well, I think I want to thank Daniel. This is just amazing work, and it's just exciting to think that we can actually achieve this level of diagnostics and cheaply. Inexpensively and non-invasively in some sense. Just drawing a little bit of blood is not as big a deal as some things you've got to go through. So I'm really, really pleased that you're doing this work, and I'm so pleased that you're part of the Baskin Engineering faculty, and I look forward to some great successes. So thank you very, very much.
Thank you. Thank you very much. Thanks, everyone.